Hello, this is Anuja Matthew. It's December 21st, 2020. Today I'd like to talk about some coronavirus variants that have been in the press recently. The main question is, does SARS coronavirus 2 mutate? And what is the meaning of the latest variants from the United Kingdom? And how does it impact transmissibility? In the last week or so, there's been quite a bit in the press suggesting that the coronavirus is mutating. Mutant coronaviruses have been detected, particularly in the United Kingdom. It set off alarms, but its importance remains unclear. In the southeast part of England, a particular variant of coronavirus has been detected at increased frequencies. And this area of England has also moved into Tier 4, which is essentially kind of like a lockdown. But what is the meaning? I want to go into the meaning of mutant viruses, particularly RNA viruses. A mutation is a change of a nucleotide in the virus genome. Most mutations in the genome do not necessarily change the encoded viral protein. So for example, if there's a change in the nucleotide sequence in the spike protein of coronavirus, that does not mean that the spike protein is now mutated or changed. Sometimes a change in the amino acid sequence or a change in the nucleotide sequence can cause a change in the amino acid sequence. For example, if you see a designation N501Y, N is an asparagine, which is an amino acid. At position 501 in the genome, that N has been changed to Y, which is now a tyrosine. So this represents a change in the amino acid sequence because of the mutation in the nucleotide in the spike protein. So even if you have a change in the amino acid sequence, that does not make a virus more pathogenic, more or less transmissible, more or less causing disease. A virus variant is a distinct virus which may have a combination of different mutations. So what we're talking about in terms of the variant found in the United Kingdom, this is a variant that is distinct, that has a unique set of combinations of mutations. A virus lineage combines the genetic information and epidemiological data. So it looks to see where these viral variants are detected in different parts of the country or different parts of the continent or different areas within a country. So tracking these lineages can be very useful for determining how a virus spreads through the community or populations. And a virus strain contains mutations that alter the function of the virus. So it might alter the pathogenicity, it might alter the virulence, it might alter the immunogenicity. So there are many more lineages of SARS coronavirus 2, but far fewer strains. There might even be only 1 or 2 strains of SARS-CoV-2 to date. Here's a table taken from an article, A Dynamic Nomenclature Proposal for SARS-CoV-2 Lineages. And what I want to point out really are there are two main lineages, A and B, and the number of genomes that are sequenced for each of the lineages is indicated here in this table, the time frame when it was sequenced, and the comments. So just as an example, the lineage A is the root of the pandemic, and many sequences have been associated with lineage A. Lineage B and the subforms of this lineage, for example, B1, largely composed sequences that were detected in the Italian outbreak. B11 contains maybe most of the major European lineages, whereas B4 was probably a lineage that was detected in the primary Iranian outbreak. So this is combining both the genomic sequence and where these virus lineages were located in the world. The COVID-19 UK GENOMICS CONSORTIUM is a consortium that sequences many genomes that have been submitted to this consortium from people who've tested positive in the United Kingdom for COVID-19. To date, they've sequenced over 100,000 genomes from people who've been infected with COVID-19. But I just wanted to mention this still represents only about 10% of the people from across the UK. So it's a subset of people from which genomes have been sequenced. And the sequenced genomes are used to identify mutations that arise and to identify which viral proteins they could affect. This is a very detailed and tough challenge to try and identify where these mutations occur, as RNA viruses mutate constantly, and how they might be meaningful. Here's a table taken from an article written by the UK GENOMICS Consortium, where they identify priority mutations that are being tracked by the consortium. So as I mentioned before, here are where the main mutations are listed. So here is a position in position 614 where there's a mutation from D to G. It belongs to lineage B1. And here, why the reasons they are tracking this particular lineage and how many sequences have been done in the United Kingdom and the time frame. So this is just to say that they are tracking multiple genomes, multiple sequences, multiple mutations, and now they're focused on a particular set of mutations that have been seen in a variant of coronavirus in the southern part of England that's causing some concern. So determining phenotype from genetic data is a fundamental challenge. And what does that mean? Just because you see a mutation in a nucleotide sequence and therefore an amino acid sequence, what does that mean for the phenotype of the protein is quite a challenge. For example, this is the spike protein, and in red are potential sites where mutations have been detected for SARS-CoV-2. If there's a mutation in the receptor binding site, so in this region, for example, then perhaps it's a major concern because this might indicate to us that if there's a mutation, perhaps then the virus binds better to the receptor and has an increased chance of getting into into a cell. But we don't know that yet. That has to be proven in the lab that this mutation caused the virus or the spike protein to increase its entry into a cell that has its receptor and at an efficient or far more efficient rate compared to the wild type or the native virus. So just because you have a mutation in an amino acid sequence doesn't mean that it actually impacts the phenotype of the virus. The variant that's causing concern belongs to the B117 lineage. It does account for an increased proportion of cases in parts of England. The cases and the number of regions reporting infections are growing. There's a large number of genetic changes in this lineage, particularly in the spike protein. And 3 of the mutations that have been described have the potential, they've not been proven yet, they have the potential to mediate biological effects. The rapid growth of this lineage indicates the need for enhanced genomic and epidemiological surveillance worldwide and lab investigations of antigenicity and infectivity. At this stage, we can't rule out the fact that some of the increased transmissibility that's been seen is entirely related to human behavior. It has nothing to do with this viral variant. The viral variant has not yet been shown to be more pathogenic, that's able to cause disease, more transmissible, contagious, or cause more disease and in terms of virulence. That will be tested. There is cause for some concern, but studies have to be done to confirm that this variant is more pathogenic. Like all viruses, the coronavirus is a shapeshifter. Some genetic changes are inconsequential and some might give it an edge. We're waiting to see that. And at this time point, there is no reason to believe that any of the mutations discussed here will affect vaccine efficacy. So thanks again for listening. This is a lecture on potential mutations and variants detected in the southern part of the United Kingdom and the cause for concern. At this point, really, I would say leave it up to the scientists to do some more detailed studies to determine how pathogenic this variant is.